scheduled, like one talk. That looks like a very stimulating afternoon. The first talk will be from Harald Hess from the Aipom Medical Institute, telling us about three-dimensional imaging of biological species.
Thank you, and I'd like to thank the Aipom organizers for a chance to be here. My path has sort of gone from, from the very pure physics orientation, originally at Bell Labs, and recently I've gone a little bit more toward the biophysical focus at Howard Hughes Medical Institute, and I think it overlaps with some of the themes here at this conference. So I'd briefly like to just follow that path and how it inspired the transition into toward a biology focus. Many years ago, in the late 80s, early 90s, scanning tunneling microscope was actually the rage, and I was drawn toward that, and also superconductivity was very, very important. So in starting at Bell Labs, I decided to focus on a low temperature scanning probe microscopes, scanning tunneling microscopes initially of various forms and shapes. And down the hallway, I had a colleague, Eric Betzik, and he was very interested in trying to go beyond the diffraction limit with a new form of an optical microscope, which was a near-field scanning optical microscope. And he had at the time imaged single molecules at room temperature with this microscope in the early 90s. So with my low temperature background in scan probe microscopy and his particular microscope technique, which was seeing well below the diffraction limit of the optical microscope, about 10 times better, we decided to do a low temperature near field super resolution microscope. And one of the first topics that we decided to go after was a gallium arsenide, aluminum gallium arsenide quantum well. And these at the time were known to have spectra which show two distinct peaks. And the distinct peaks were often associated with what might have been monolayer thickness variations in the underlying quantum well material. For example, it could be 11 monolayers thick or 10 monolayers, and you'd get either a red or a blue shifted spectrum. And we were then able to image that, for example, in how that varied in space. That was interesting for the technology in X and Y. Now, we actually had a surprise when we looked at it very carefully at the spectra. They really weren't two broad peaks, but instead there were lots of very, very fine, distinct peaks in there. So with that, we realized we were looking at the individual quantum constituents of the fluorescence. It was a big surprise to us. So essentially, if we're looking here in a quantum well, an exciton, electron hole pair, could get stuck, localized, and then it would have its own particular environment, its own energy levels, and then we would see this at one very specific wavelength. It would light up, let's say, as a little red circle. So you could actually localize where that light was coming from, even to sizes much smaller than that circle. So the whole data set would look like an X and Y if we scan a full area, and then the third dimension could be wavelength. So we have these volume data sets like this, and each one of these little donuts, which sits here in space, is one energy state at one particular site in the system. So that inspired Eric later on to think, well, gosh, maybe this is a more general principle that one could use. If you spread out some data, which together might all be blurred together, but spread out in some third dimension, you can actually localize where the exact point of the emission is from, replace it with a dot, and add them all up, and you can form what could be a higher resolution image. Unfortunately, there really weren't any ways to do this conveniently, which was useful for biology in wavelength. These were just too spread out for the biological fluorophores at the time, and that was sort of pretty much it. And at the time, Bell Labs was having an increasing focus on on the technology to make it relevant to the economy. And Eric was getting a little bit uncomfortable with the limits of the near-field microscope and left. And I likewise was also getting a little bit uncomfortable with the limits of low-temperature scanning probe microscopes, particularly in how it could impact the technology, which was very much the focus of Bell Labs at the time. So we both left, and we both decided to work for test instrumentation for industry. He went off to Michigan to try to revolutionize the auto industry and prevent ultimate bankruptcy, and 10 years later failed miserably. I went off to California to get rich in one of these fast-growing new companies and failed miserably. I started out trying to, there's a little company called Phase Metrics, which was supposed to revolutionize the hard disk drive industry. Basically, they started firing people as soon as I joined, and developed various forms of multi-phase interferometry. I thought, well, maybe we can do nanometer measurements very quickly, and that's useful, but essentially all our customers were bankrupt, so they didn't want to buy anything anyway. Later on, that little company was acquired by a much bigger giant, KLA-Tencor, which actually built some very interesting high-throughput measurement instruments used to measure masks or wafers. So you need to do nanometer measurements at gigahertz rates, and actually, I was actually philosophically drawn to that whole concept. It's not only nanotechnology, there's also trying to deal with vast volumes of data, and industry really makes a special effort out of it. So this little machine you see down here, that's a $40 million microscope that basically inspects masks that print the computer chips. At some point, I became a little bit frustrated, and Eric was also failed in his attempts, and independently we both decided to move on, which means freedom and something else. And so we have the inevitable question, what now? So, we teamed up again, met in some national parks, walked around trying to find answers to where in the future science could be, and we were walking a lot. I tried to pretend to be, I was gainfully employed, formed my little shell company called New Quest Research. He was New Millennium Research, and we tried to figure out what to do. So, one of the things that we learned about was this thing. This is the green fluorescent protein. And this is a label. It's a genetically encoded label that was derived from the jellyfish, and people learned how to isolate it, make it, and attach it to an arbitrary protein of your choice in any biological material, in cells, in creatures, in anything. So you can have literally a light bulb on any protein of interest. And that was just being used widely. For example, here are some cells where you can label the mitochondria or the Golgi or different parts of it very nicely. And in our searching to figure out where to go, Eric had one microscope concept, and of course to find a biologist, we went to the National Magnet Lab, and there was this person, Mike Davidson, a very remarkable person, very unusual to be in the Magnet Lab, but he explained to us that there are actually a new category of fluorescent proteins called photoactivatable fluorescent proteins. So this one, it might glow green, you shine it with blue light, and you can turn it on red. So they just don't glow, you can actually switch them. You can turn them on and off at will. And knowing that it sort of dawned on us a few hours later that if you have a light bulb, a label, that you can sort of turn on and off at will, it actually is a missing link that can combine with this earlier idea that came from our quantum well experiment in Eric's little paper, that you can now make a higher resolution microscope. So here's a little demo of it. If you have just isolated molecules light up, you can localize the center of that spot very precisely, and you just write those points that are localized, and in the end you can resolve something that in a regular microscope, if everything glows together at once, would be a blur, but here you can get higher resolution. So that's the palm microscope. And that we sort of, oops, have to get this off here. And with that idea, we went to work. Here's my living room, and there's the optical table, and so Eric flew over, there's the computer center, and we started, within a few weeks, put something together, no granting agencies or anything like that, so we could move quickly. And a couple months later, we connected to one of the inventors of this photoactivatable green fluorescent protein, and a real excellent cell biologist, Jennifer Lippincott-Schwartz at NIH. We put the scope into a suitcase, brought it out there. She had a dark room. We hung out there for a while, labeled some lysosomes in a cell in a thin section, did the blinking, and voila, we got high resolution images. And a little bit later, we got employed. This is Janelia Farms as part of the Howard Hughes Medical Institute, and they had just broken ground on this new institute. And the goals there were to develop new imaging technologies and also to understand neural circuits. And so we went on. My initial project at Janelia was trying to think, well, can we take this three-dimensional imaging technology and, or actually this palm technology, and extend it to a third dimension? And having done some interferometry at industry just a little bit earlier, I thought, well, this is a good place for it, and developed a microscope technique, which we call Aipom. So you can think of this as a grand two-slit interference experiment. Essentially, you have two objectives, one above, one below, and there's a little molecule sitting in between, like right there, and it's radiating like crazy. And the photon goes up and down at the same time and can interfere in that beam splitter. But that's not just a simple beam splitter, it's a three-way beam splitter. So we get not just one phase and the 180 degrees, but three separate 120-degree phases. So if the molecule is a little bit higher or a little bit lower, you'll see the light goes from camera 1 to camera 2 to camera 3, and the ratio of the different images, of light in the different images, really reflects how high that molecule is in the z direction. So we can read out that position to tens of nanometers resolution. And so that takes optical resolution from what would normally be the case for a confocal microscope, where you have a diffraction limit of maybe 250 nanometers, half a micron maybe in the vertical direction, to a point spread function that's about 10x smaller, like the little lentil thing down here in the corner. And so we built that and started doing a few example demonstration imagings. Here's yeast, which is budding. As it buds, there's a protein called Septon, which we could label with these fluorescent proteins. And for example, here you can just sort of see the details of that structure. Here's another example. This morning actually saw some focal adhesions. Well, this is a focal adhesion protein, integrin, and this is now seen in palm microscopy and now Aipom, where the color code refers to how high it is above a surface. And here you can get a sense of a vertical height. The yellow part, that's where it just is adhering to the cover glass. And the tubular purple and green tubes, that's the endoplasmic reticulum, which is just sort of floating a little bit above that part in the lamellipodia here. And we sort of really rely on biologists to come in and give direction to this. Claire Waterman, and an excellent student, came in and really wanted to disentangle what is the nature of these focal adhesions. So the focal adhesions are places where the cell tries to attach itself to the outside world, or here to the cover glass, and it forms these spots. And that's a collection of about 100-odd different proteins which anchor themselves, but it wasn't quite sure what the organization is of that. But by going in, measuring the height of the different proteins, some connect to the actin, some connect to the integrin at the bottom, some are regulatory proteins, and we can actually form you know, a little cartoon of how it's organized. And here's another example. Another colleague from NIH, they wanted to understand where an escort protein is located. This is involved in HIV. So here's a HIV virus. It has a little capsid consisting a lot of gag proteins, which we can light up in red. And there was an open question of, does this escort protein sit down below, underneath this budding virus, or does it sit up in the neck? And so by labeling these escort proteins, we could then see, well, it looks like it's really almost enclosed by this capsule. So we have a model where these escort proteins are involved directly in the pinch-off process inside the neck. So this technology matured, and it's there's not that much technical development on it, but there's a lot of development that still needs to be done in terms of preparing samples and making it an easier and easier tool for biologists to use. So there's an advanced imaging center that's now supporting that. And then I've turned a lot of my focus now to electron microscopy, and I'd like to give you a few examples of where we're going on that. Now, when I joined Janelia, actually I resonated with this particular project. Before I was in the semiconductor industry, and you'd like to image computer chips. My boss at Janelia, he was very excited about fruit flies and trying to understand their brains. So whether it's a fruit fly brain or a computer chip, it really has a lot of tiny, tiny little details and a net complexity which are very comparable. You know, you think of millions of transistors, you can think of you know, tens of millions of synapses and very tiny wiring going in there. So the data analysis challenge is huge. If you just take this little fly brain, it's about 1/3 of a millimeter, and in order to see any usable detail on it, you have to chop it up to about 8 nanometer voxels. And you just sort of cube this ratio, and you get, you know, close to 1/10 of a petabyte of data. And at 1 megahertz acquisition rate, you have to go for about a few years until you have the full fly brain. So it's a big acquisition job, but it's actually even a much, much bigger analysis job. So this is a little bit, from Kristen Harris, a little bit of a video just explaining a little bit what the standard approach was initially for doing this. So you just take your sample. The sample is actually stained with metal, which sort of lights up all the membranes with heavy metals. And so when you take an electron microscope picture, you get this pattern. You have a computer try to colorize things in, lots of humans to correct all the computer errors. And then you take that data, stack it up, and cross-connect, register, and then you have the beginnings of the picture of what the connectivity is, where synapses exist, and things like that. So the question, when I was thinking of doing this for the fly, was trying to understand, well, what's the best way to go around, attack the problem? Really, the biggest problem is the reconstruction, the data analysis. So one's faced with a number of possibilities. You already saw this. You cut the sample into sections and do transmission through it. Or you can put these sections onto a tape and look at the backscattered electrons. Another colleague went for DINK, developed a technique where you cut the surface with a diamond knife and look at the block face, but still looking at backscattered electrons. And I opted for this last approach, and I'm going to talk about this in a little bit more detail. Here, we abrade the surface with a gallium beam, ion beam, and we can take off just a few nanometers at a time. So here's a sample. We image the surface, and then we shoot ions across, image the surface, abrade away a few nanometers, image it again, and this goes on, a 1-minute cycle time for each cycle. So over a period of days, you munch away on the sample and you collect a 3-dimensional data stack. And the big advantage and reason for going this is that the Z resolution is the best of all the various options. We can just ablate away only 8 nanometers or 4 nanometers, whatever we want at a time, and get relatively good Z resolution, whereas if you have to cut sections, things get choppy and you might not be able to see the same level of detail that you'd otherwise see. And that can really get in the way of reconstructing and interpreting the data. Furthermore, synapses were very clear. They stood out in the density on this, and finally having a block face makes registration. You don't have to sort of align and register different cut sheets very much, so that makes that problem easy. So again, just to reiterate, we're going to take a little more time in imaging, but hopefully we can take this complete reconstruction time down. Notice the vertical scale. This is talking about millennia, and ideally you'd like to do an experiment in a few of your lifetimes. But there's one problem. The FIB-SIM can't acquire relevant volumes. So if you run the machine and you start taking data at that resolution, you got a volume that's about the size of that little green speck. It's just a fraction of a neural circuit. So there's some problems. The machine can't run more than a few days at a time. Essentially, the gallium source has to be refreshed. There can be temperature fluctuations in the room. Restarting the machine is not trivial because you want to go in there and keep going right at the nanometer level where you left off. If you lose data, you lose a big chunk of the connectivity, and what might have been like weeks or months worth of data acquisition are just lost. So it's very unforgiving. So we spent some time addressing that, pausing it on any sign of trouble, combining two companies, FEI with the FIB column, Zeiss with their SEM column. We call the thing VICE, and integrated other features, for example, feeding back on the gallium beam so it touches the sample at just the right level, not too much, not too little if there are some other excursions that take place. And after a few years of this, it allowed us to expand the volume size, and now beginning to get to relevant volumes. This little volume took us about 3 weeks to acquire, and it took another 2 years to reconstruct. And this is now part of the optical lobe. The fly have little omnitidia, and there are 7 little computational modules that are in there in that volume. And this is now dense reconstruction, and we're trying to sort of speed that process more and more. Here's another example. This is the something called the mushroom body. Fly has an object which basically is associated with olfactory learning. And so a random code of neurons which should describe some olfactory stimulus can be written in and define an output behavior. And and that learning takes place in here. So we've recently, again with a couple of years' effort, reconstructed that and trying to understand the rules of what the wiring is really like in these systems. And we continue to expand. This is now getting the whole optic lobe. This is now 100 microns in size, and you can see 3 big computational units here. This is the medulla, the lobula, and the lobular plate at the bottom. This is about 150 microns across. This thing took us about 3 months, 100 days to acquire. And again, the retina's out on the left side. And so one of the main goals that they got, or trying to validate with this kind of data and previous data sets, is like, how does the fly detect motion? So you have to detect differences between the different omnitidia. There's one time retarded to the other, for an off signal, for an on signal. Try to identify, is this a threatening thing or not? Let me see if I can just sort of stop it right here. This is about 10x closer focus. And if you look here, these dark things are mitochondria. You might see some dark spots, like right in here, these things. Those are synapses. That's actually where essentially the transistor is located. There are several of them in here. Like down here is a little dark one, mitochondria. If you look, you might see little lengthwise tubes in here, little dots, those are microtubules. Now the trick is, when you're reconstructing, you have to sort of identify every one of these tiny little processes and connect them up into a big long network, which is your neural system. And fortunately, that's the job of another group, not me. So we have an independent team working on that. So, okay, so we're up to 100 microns, but there's a limit with FibSim. Essentially, if you try to mill across a surface that's longer than 100 microns, the beam goes unstable. The milling's not uniform. So we needed another approach. And Kim Hayworth, who joined my group, actually proposed something which he calls the hot knife. And the hot knife is you just basically take a diamond knife, heat it up, add oil, and cut. And so if you do about 10 or a couple dozen cuts on a plasticized fly brain, which has been all prepared for EM, you can make a couple dozen little tabs, 20 microns thick each, laminate them in a little bit of plastic, and spread them out on a few of these focused ion beam SEMs. And so now, because the thickness now is 20 microns, way less than 100 microns, we're able to access the whole fly brain. But the big question is now, well, how well can you stitch these surfaces? Are they good enough that you can really follow a nerve from one over to the other side? And it turns out the answer is they're pretty good. And you can trace them with about 98% reliability across that interface. So presently, we're actually on our way to trying to do FibSim of the whole fly brain, and right now we're focusing right in here. This part here is called the central complex. We already have 4 tabs completed. Central complex, like you see right there, it has a little something called the ellipsoid body, the fan-shaped body. This is actually with the compass of the fly. It knows its orientation, and if you look at certain cells, they light up depending on which way or what it sees in spatial directions. And so this data will then be combined with the electrophysiology data and people who are expert in this field to figure out what's going on. And here's some example of a couple of the data sets of the tabs. There's a cross-section of the fly. This is a little bit of close-up. And yeah, I think, if this is complex matter, I think it is. There's just an amazing amount of wiring going on, synapses throughout this whole thing. The synapses, sometimes they're fan in, many dendrites come into one place with one output, or sometimes it's fan out. One comes in and several go out. Just a variety of structures in there. And here's just another close-up of the same thing. So if we stop, and again, you can sort of identify some, the little dark areas like that one here. You get good at this after a while. These are synapses. And the whole key is the data has to be good enough that it could be done as much as possible with automatic reconstruction. Otherwise, we're just filling up hard disk drives.
Sorry, just one question. You said that the small black dots are microtubules there?
Yeah, if you look, for example, you might see little lines. Sometimes they're ER, but very often they're microtubules. And the little black splotches that you might see here and here, those are synapses. They're often like many things come together in one little converging point. Yeah, this is different from mammalian synapses, where you usually have just one nerve synapsing onto the other one, very simple. Here it's like 1 to n or n to 1. So, oops, okay. So with that in mind, we decide, well, there's maybe one more thing we can do. We can shoot for resolution. Before we're shooting for speed to get through the full fly brain and get some volume. Now, we can go in the other direction. We can lower our energy, and when electrons go into the surface, they might spread out less than at our high throughput speed, so we can get better spot size. Or we can drop our aperture. In an electron microscope, if you go for a smaller aperture, you actually get better resolution. Turns out it also means things slow way down, and you just can't image that much anymore. But the fact that our microscopes can be run now for years on end without interruption means we can go down here, get maybe about 2x better resolution, and run for a long time and get maybe volumes which are interest, let's say, not as big as a cell, as a fly brain, but maybe perfectly good for cellular type imaging. So here's an example of low resolution and higher resolution, and the imaging speeds are different between the two. And I'll just give you a few pretty pictures from here on out. This is the Chlamydomonas, one-cell organisms, low resolution, high resolution. Here's a movie. The movie is going through in Z thickness. Here you can see some, that's a nuclear envelope right in there. You're going to see the little cilium appear there. This is going to start, this is a close-up of the cilium and the molecular motor that's driving it. And, on the cilium? Okay, I can just pause it right here. So there you can see there's about little microtubule pairs, about 9 microtubule pairs in that arrangement right there. Or if we go a little bit further, you'll see the second cilium sort of appear down here and has a little pinwheel type structure as it comes out. And all sorts of funny anchoring at the base. And again, just to play around, here's the nuclear envelope. There's a ton of data here, but let's just mask out the thin outer shell just around the outer part of that nucleus. And you see some little white spots on the outside. This is the same data set as before. And you see this. So this is the surface of the nuclear envelope, and these little pearl necklace structures, these are polyribosomes. So this is where there's a little RNA sort of probably along these structures, and each one of the polyribosomes is manufacturing its own protein of choice. In the middle here, for example, right there, this is a nuclear pore that's right in between. This is seen in regular EM, but the trick is now we're seeing this in 3D. No real constraints. We're not limited to a particular section. Here's another surface of the nucleus. You can see all the little holes. Those are the nuclear pores. Here's an example of a neuropil. I just missed it, but there's a Golgi in there. If you reorient it in the right way, you can see the Golgi apparatus is actually constrained by a filament that goes around it. The ability of having a three-dimensional data set that you can sort of rotate and manipulate, allows a lot of access. Another colleague was just, again, on the same data set, just segmenting out one dendrite. You can segment out the endoplasmic reticula, you can segment out the mitochondria, look where it touches the membrane since they're associated with lipid exchange, set up the details of synapses. So to summarize, FibSim has certain advantage. You get nice 3D volumes, larger volumes of interest, for example, a whole cell, and you can look at it at arbitrary angle. And one final little plug, we can now combine the high-resolution light microscopy with electron microscopy. Light microscopy gives you protein-specific information, but usually doesn't have good context. Electron microscopy gives you great context, and it's always a challenge of sample prep on how to put the two together. And here's one example that we did earlier. FibSemming here. The orange color is where the DNA, the mitochondrial DNA, sits within a mitochondria. And for the future, I think we're actually in a pretty rich space right now of, I think we're a little bit back to the past. I sort of feel like it's a little Cambrian era where there are just so many options, so many protocols to choose from, so many problems to choose from. So I don't know what the future is, but that's actually for many of us to sort of have fun with. And of course, I'd like to thank a lot of people. Eric for sharing a little period of unemployment and as a great colleague, present lab group, and then of course many, many collaborators who sort of give meaning and direction to all of the biology that comes from here. So thanks.
So I think that one of the conversations that resulted from the Obama Brain Project was that suppose you really can do everything that was claimed with the mapping of brains and neurons and this and that, would you then actually be able to answer the scientific questions that you had when you started out to do that project? And as you were showing the fly brain stuff, I had the same question came to my mind. And this is probably not the division of labor that you're involved in, but still you hang around people who must worry about this kind of stuff, and I wondered what perspective you have.
That's the kind of question that gives me nightmares at night. I really worry that, you know, we can collect the data and we can get connectivity, but it's only going to be a partial answer of many things. Even from the little pieces that we've gotten, we might get little clues of some little subcircuits of, let's say, of the fly brain, of how the visual system works. Exactly what turns on, what's associated with memory. Is it visible with an electron microscope picture? Do we need more in there to see which proteins are there or not there? I think there's a whole host of unanswered questions that still need to be considered seriously. I think it's once you get this data, it's not like, you know, here's the Hamiltonian of the universe, and you have all these endorphins in your brain and are really happy. This is maybe just the beginning, and people can use a data set as a reference for where they're doing maybe painful experiments on particular nerve cells and what happens with certain environments and behaviors. So I think it's only, it's a beginning, and it's going to be a long path. I think later on maybe correlation, trying to understand the details in its greater entirety is still yet to be done. And I think a lot's going to come from the ground up too, from trying to understand how do these nerves decide to know where and how to wire and make the synapse, what proteins get expressed, why is actin shooting off in this direction, the whole feedbacks. And it's complex. I mean, what can I say? I'm not pretending this is going to be one of the answers, but I think it's hopefully one of the pieces of the big puzzle.
I'm not sure if I get it correctly, but for the brain study, you make slices of the brain and then take a picture. So it's something that it's not actually in the brain. I mean, you can just do it live. For example, if you have a rat, so can you just immobilize that rat and then look at the brain, for example, see the responses under different conditions?
Right. Yeah, there are a whole suite of approaches which are going after the problem. This stuff here is very, very dead. But for people do live imaging, the probes that people are developing, just in the past few years, they can light up on calcium spikes, and you can begin to see neural activity, or begin to get a little bit of a picture of what's going on, but not at the detailed high resolution level. These brains, we have professional fly brain dissectors, and they take out the little fly brain and fix it, extract the water out, stain it with heavy metals, so all the membranes get stained, and then re-infiltrated with epoxy, and then trimmed up. So those little pieces of plastic of fly brain basically reflect all the membranes of the fly brain itself, and then we're just sort of looking at that, sort of layer by layer by layer as we sort of gradually mill through it. But again, it doesn't show things acting in sort of a live mode. The movies, they're not live movies, they're movies of just going in Z depth.
Thank you.
I wanted to know what is the time to process one layer to construct just one layer?
Okay, let's see. There are probably some numbers I could give. Usually we process it by volume, and let's see, I think a 10 by 10 by 10 micron size volume is going to be a few days or a week or somewhere in there. So once you get things, which turn into about 1 million cubic microns, we're very quickly talking with a team of 10 people, a couple of years. And we're trying to sort of, improve the algorithms, improve the data, and soon it's going to be developing into a technique like, well, you can get it 50% accurate if you spend this much time. If you want to get it 90% accurate, you might have to get an extra order of magnitude in time to process it. And so there's sort of a tuning that goes on exactly how accurate you really want to be in trying to understand the connectivity and what's needed. And that's even an open, fair question in itself.
Are you worried that the ion beam might be damaging or distorting the surface that you are imaging? And also, learning that it takes so long to do one sample, is there a lot of sample-to-sample variability, if you have any idea of that? If you lose a piece from one fly, can you replace it from a piece from another fly and redo this?
Okay. Yeah, let's see, we definitely destroy the sample as we're milling it. I think the ion beam, so it ablates away. That sample, those few nanometers are off on the wall of the chamber. And then the next one, I think it's relatively pristine. There might be a little bit gallium ion embedding. It might just sort of improve the conductivity, but it doesn't really change the structure. I don't think really would form any artifacts. That would get in the way of imaging. And oh gosh, the second half of the question was
sample to sample.
Oh, sample to sample. Yeah, that's a very good question. How much difference is there between two flies? We try to make everything one fly, so that the variation isn't there, but that puts big demands on perfection. No mistakes. If we miscut or destroy one of those tabs, it's all over. We do have a little sense of variation when we did an earlier experiment on the fly on the tibia. This is like a circuit which repeats itself for every little islet and next one and next one. So you can ask the question, how much variation is between one column that serves that little, part of the compound eye versus the next one over? And you can identify certain neurons and say, okay, this one typically, this type A connects to type B with about 100 synapses. And you look on the next one over, how much connectivity is there? And it might be a 110. So there is a variation, but the variation is square root of n. It sort of almost falls a little Poisson law. And, which you, maybe naively, it's not that unreasonable. But, beyond that, you want to get then soon to the next level of experiments of, well, what can be taught? How does the memory look like? What are systematic differences in, the experience of one fly versus another one, or learning that's taking place, or growth stages? So this kind of stuff is, I think, just a little bit the beginning of where you begin to ask questions.
Okay, are there any more questions?
Maybe just
Oh, yeah.
this isn't your thing, Harald, but do you have any comments on whether, so there was a report of using ND diamond to look for activity, to actually monitor activity on an individual neuron, and that gives you resolution, although the problem is how do you get all of those ND diamond centers into more than one neuron. But do you think there's any future in that, or is that something you don't look that much?
I haven't personally looked at it, but I think my standard response is trying to get some of the new materials, whether it's quantum dots or NV diamond, integrated into a biological system is a huge problem. It's just not natural. I mean, even we worry about having, a genetic label, like green fluorescent protein in there. You say, well, gosh, did that sort of mess up with the structure that you're hoping to try to confirm or validate? So I think it's interesting, you have some very nice sensitivities, but you have a huge problem of delivery and how widely usable is it. And the progress on some of these other fluorescent probes is amazing. So it's always, choice of competitive technologies for, the goal at hand.
Okay, let's thank our speaker one more time.